I'm going to be recording this presentation so that we can archive it on our website. So both the recording and the presentation will be available. This is part of a series that we're doing with regard to registries, part of Genetic Alliance's boot camp, which guides advocacy organizations and community groups through the process of starting and sustaining a registry. We're delighted today to have Dr. Woody Gandy with us. Woody is from the Ehlers-Danlos Society, and he is both a father of a young woman with Ehlers-Danlos as well as a researcher. And Woody's going to share with us information today about his HEDGE study. Without further ado, Woody, it's yours.
Great, thank you, Sharon. I'm glad to be here, and I must say that as I've become involved with the Ehlers-Danlos Society and then with Genetic Alliance, I've become more aware of the broader community of people involved in genetic conditions. I really feel that I'm part of a broader community, and that we all have in common the challenges in sort of getting to the root of some of these very perplexing problems. Not only sometimes in our case, even finding the causative variants or what might be going on to give rise to this, but how it can give rise to the phenotype in its various forms. So I'm glad you all are here. I'm very gratified that we had such a nice turnout. I hope this will be useful to you. You can see that the logo for the Ehlers-Danlos Society is this zebra. That's because in the Ehlers-Danlos community, people who have this condition are often not diagnosed for a long time, typically 14 years, that's a typical length of time. And my daughter was similar to that. She didn't have a lot of problems as a child, but as a teenager, she began to have some odd things going on. And I have to tell you, when I was in medical school, I heard of Ehlers-Danlos Society. We spent about 3 minutes on it in one lecture. And I did remember that it existed. Didn't quite remember much about it, but I didn't think about it with regard to my daughter. So here you'd think if the dad's a physician, you'd have maybe a better chance of cluing into what's going on. So imagine for all the people who don't have that in their family, it's going to be very difficult. As a teenager, she was running cross-country track and began to have pain problems, particularly low back pain that was unremitting and quite disabling. And we saw various specialists trying to figure out what was going on. And it's pretty unusual that a teenager is going to go to the extent of having bone scans and the extent of evaluations she had, and just never quite solved it. But for 2 or 3 years, she went through a period of quite a bit of pain related to that. And then later, as she entered in her 20s, she encountered issues with gastrointestinal problems, with difficulty with motility, nausea, abdominal pain, pelvic pain, and continued to have broader pain syndromes: neck, back, hips, shoulders, trouble sleeping, can't sleep on her side because shoulder pain, then developed sleep apnea, and other complications. She has Hashimoto's thyroiditis, which is a known complication as well. People with this problem, with the hypermobile Ehlers-Danlos syndrome, are very flexible, and but it's not a normal flexibility because they develop a lot of problems with it. So, see if I can make this slide go forward. There we go. Okay. There are 14 types of connective tissue disorders called Ehlers-Danlos syndrome in this large umbrella, but 11 are very, very rare. We're talking about 20 or 30 people. There are 3 types that are less rare, and these are the ones we hear most about. There's one called the vascular Ehlers-Danlos syndrome, which gives rise to vascular complications and tragic early deaths in some individuals. Classic Ehlers-Danlos syndrome has been well recognized and genetically defined for some time, and these folks have very flexible skin. And then the most common by far is the hypermobile Ehlers-Danlos syndrome, which is undefined. Of all these types, the one that's the most common is the one that is not, we've failed so far to find a genetic cause for it, to understand why people have these this phenotype. We don't know quite how frequent it is, but we think it's more frequent than is possibly recognized. It's hard to do these kinds of studies. It's typically thought of with the more stringent criteria that came out in 2017 as being about 1 in 5,000, which makes it a rare disease. But it may be much more common than that, at least in milder versions. And some people have estimated that it may be as much as 1% of the population. But I don't think 1% have the degree of problems, for example, that my daughter has. So it's really a matter of severity. And she's probably about average. There are people with much worse problems, and I'm fortunate that she's got a lot of coping skills and is a very strong individual, so she deals with it. Now, we see that the what we've observed is it appears to follow an autosomal dominant inheritance pattern. And that's just what it looks like, but we haven't found a gene that is an autosomal dominant gene. Now, the reason we use this zebra logo is because in medical school, you're taught that not to look for zebras. If you hear hoofbeats, horses are common, look first for the horses. But this is a reminder that there are zebras, and people do have long delayed diagnoses before they're discovered. And during that time, they're often treated as having some sort of psychological problem, which further traumatizes them. Now, these folks are very flexible, have this problem of chronic pain, dislocations, and subluxations, and what are called comorbidities. And outside of this EDS area, comorbidities typically means unrelated problems. So someone has congestive heart failure, it's totally unrelated to whatever their main problem is. But in this circumstance, we use the term comorbidities to describe these other syndromes that we see in conjunction with hypermobile Ehlers-Danlos syndrome, such as postural orthostatic hypotension syndrome, these GI disorders, mast cell activation syndrome, where they have anaphylactic-like reactions and a lot of rashes, throat swelling. They have fatigue and other issues. So this condition was sort of vaguely defined for a long time, and a lot of people could fall into it. And so for the purposes of research, the decision was made to kind of try to narrow this down and see if we could come up with something a little more stringent and defined. So in 2017, the new criteria came out, and they're really based around 3 clinical findings. You have to have all 3. The person has to be hyperflexible, and that is measured by the Beighton score relative to their age. Although we do also give a little leeway to historical evidence of hypermobility from childhood. And there's a set of 5 questions we can use to sort of modify the criteria for meeting this first criterion 1. The second criteria has to do with finding other characteristic phenotypical findings. And you need at least 5 of 12, or you need a family member who has that. If you don't, but you have someone else in your family who fulfills this definition, then that will suffice. And you need the chronic pain and or dislocations. And so for criterion 2, we need at least 2 of those features. Criteria 3 is basically just to rule out another cause of the condition, particularly other genetic causes. Well, so I joined the board of the Ehlers-Danlos Society. It's been going on 3 years now, and wanted to become involved to help further this goal of defining the genetic cause. And I'm under no illusion that that's going to just give rise to some immediate cure to the problem, but I think it's a step that has to happen for us to understand the pathophysiology and to be able to move toward some effective treatments, specific treatments. So what we're doing is a population study of 1,000 participants with HEDS, that's hypermobile EDS. So we need to collect samples, and so far we've stuck with blood, perform whole genome sequencing compared to a control group, and then look at the 20,000 genes and their associated regulatory regions to try to identify a statistically increased number of mutations in some area of the genome. And we picked 1,000 participants based on a statistical analysis that Joel Hirschhorn at Harvard did and published some time ago. And he had HEDS particularly in mind, trying to kind of understand what size would be a good target. And what we know is that this has to be more than one genetic variant, because there's been enough work done that we would have found that. So by looking for 1,000, having a population of 1,000 participants, we could detect even 30, 40 or more variants that collectively cause this syndrome. Now, the challenge often in genetic analysis is that only a small part of the genome codes for proteins. So when we look at the coding part of the genome and the known regulatory areas associated with it, we're still only looking at a small part of the genome. So it's possible we may discover that we come up empty-handed, and they have to say, well, now could it be something in what's known as the dark areas of the genome? Could it be epigenetic? There's still a lot to answer to try to understand about this. But whole genome sequencing has a real advantage over exome sequencing, because we can look at things like splice sites, regulatory areas that would not be shown in in an exome sequencing. So to get moving with this, we thought about how we might go about it, and we're a little bit daunted by the idea of enlisting a multiplicity of different IRBs and institutions and recruiting around the world, and we know how costly all that can be, and how much, for a small organization, how challenging it can be to put that together. So we thought about our situation, and we realized that we have a very strong connection with our community. We have a mailing list of 60,000 plus people, and presence around the world, with good connections with scientists. And as of the last, just the last several years, the society had become much more active in outreach and holding conferences, educational conferences. And we thought we might just be able to do this by working through an independent IRB, we chose Genetic Alliance, and holding events outside of specific institutions, reaching directly to our community and recruiting people that way. So now what we found is that, with the Genetic Alliance IRB, we don't really, our clinicians don't feel that they need to have a separate, different IRB approval to come and volunteer at these screening events where we recruit participants to the study. But they do feel that they're required to obtain local IRB approval to get into their medical records, to use their contact information to reach out to their patients, obtain consents at their facilities, or draw blood at their offices. And also the clinicians who are doing the analysis have told us that they're going to need to get their local IRB approval. Well, that's fine, because fortunately there is this system now of IRB cooperation, and we're not too put off by that. But we got moving with this project, doing it through our sort of independent approach, holding these conferences, holding these events to screen people at conferences, in conjunction with conferences, using volunteers. So we're facing a number of challenges in doing this kind of work. First, the diagnostic criteria. In our case, because we have lots of people out there who have been told they have hypermobile EDS, but not under the 2017 criteria, we can't just rely on that diagnosis. And we have to confirm that they really meet the 2017 criteria. And most don't, because they are stricter than what was used before. So that's a challenge. And it's a little bit of a challenge in the PR aspect too, because we have to be careful. When these criteria came out, some people felt they were being excluded or minimized by being put into the category of hypermobile spectrum disorder, which is what we use now for those that don't quite have all the criteria. Personally, I think it's all the same condition. As I've seen these people, they all look the same, and it seems pretty arbitrary to me. But I do understand the need to try to narrow the definition, because this condition overlaps with others like fibromyalgia, and so it can be a little difficult to know that you have a very specific connective tissue disorder. We have the challenge of outreach to these people. How do we reach them? How do we recruit them? And we used social media, and we found that it was very effective, and we had people clamoring to become involved. So it wasn't too hard. Now the pre-screening, that's where the registry came into play, because what we wanted to do, if we're going to have these events, we need to use our precious slots for screening effectively. And our goal would be to have almost everybody who shows up to be screened turn out to meet the criteria. So we're not wasting people's time, both the clinicians and the people who may fly across. We had people fly across Europe to an event in Madrid, for example, and elsewhere. So we don't want to waste anybody's time. So pre-screening to try to get a handle on whether they really meet these 2017 criteria has been important. Then, of course, we have to find a way to collect the sample. Now, that has been a simple matter of just doing it at the screening event, but I'll talk about the new complications there, as you can imagine. Since this is an international project, we have to ship those samples. So we had to learn the complications involved there and find the right partners to do that. And then, of course, to find the right data platform, we're still in the process of doing that. We haven't actually sequenced these things yet, so we're still in the collection phase and the analysis. So how does the registry play into this? So the first thing we did was to create the global EDS and HSD registry. It asks a fairly detailed set of questions. I look back now and I think we were maybe a bit ambitious in believing that we could really guide everybody through a detailed self-clinical exam. But still, I think it's pretty good. So we try to guide them through all the findings and get them to do a self-assessment of what findings they do have. And we use that for our pre-screening. So we now have about 10,000, I think it's about 9,500. So I'm being a little forward looking at the 10,000 number. Most don't quite meet the criteria. But may have very severe symptoms. So the basic, what we found in our case for our condition, the main things we look at to determine whether they are likely to pass is the self-reported Beighton score, and then these 5 questions about flexibility during childhood. You know, were they able to perform tricks? Did they have dislocations and things like that? And then we ask about these findings that we need to reach the criteria 2A, these phenotype findings, chronic pain, and we look for findings suggestive of other syndromes. Now, generally, if a physician has seen the patient and has diagnosed HEDS, we may be able to just work off their medical records and say the diagnosis is adequate. But I'll talk about that here in just a moment, a little bit more detail. Of course, they have to be interested in being in the study. So the pre-screening process is that, you know, these folks go to the registry because we've reached out through our social media efforts, and they've gone and completed a very detailed registry entry. We have 2 surveys. One is a personal history survey. Another is a family history survey. And turns out we're not really using our family history survey as much as we thought we might. We may still do it later, but it's a little bit subjective, because when someone says their cousin has it, we don't really know if they meet the 2017 criteria. So it gets tough. So these folks are in the registry. We, of course, export the registry file periodically, and we run through a processing that we devised. We just work right within Excel, and we have some Visual Basic code we use under the current program. We have algorithms that analyze their answers, compute scores, determine whether they meet these different aspects of the criteria, and we define a cutoff that has largely been very much related to this category 2A, these phenotype findings. And what we found is that, you know, if they self-report 3 of them, we probably will find 5. And so we've kind of learned where the cutoff can be to have a high rate of pass when they show up at an actual event. We then, based on these criteria, will invite them to an event. Now, previously, we would focus on a specific event and invite people to that specific event. And so we have a web page that they can go to where they indicate their interest in being part of HEDGE. They give us their email address and their name. We match that to the registry, and based on those expressions of interest, we would pre-screen them and invite them to a specific event. And when they indicate their interest, they tell us which events that are coming up they're interested in. Well, we found a better solution than that. That was a lot of back and forth. That was cumbersome. And so we ended up implementing an appointment system that would be like you'd have in a doctor's office. It's an add-in it's on the web for them. And so now what we do is we look at the registry, we analyze the people in the registry, because they have to be in the registry. That's a requirement. We required that they do the registry to be part of the study. So they're going to have to be in the registry any way you cut it. So we look at the registry now, and we determine who we want to make an invitation to. And we then just let them go and find their own event, their own appointment, and set up an appointment for themselves to come to one of these events. So that turned out to be a simpler method. I definitely recommend that to everybody. And one of the problems we got ourselves into with having this separate information flow coming from a web form was that we'd have them come there and they'd give us a different email address than they used in the registry. They would use a nickname or a maiden name, and we'd have to kind of ferret out whether this is the same person or not. And so we kind of struggled with that. So I will say, if you get into this, be careful about having more than one flow of information that doesn't have a common unique identifier. That's more of a artifact of how we did it, and with the current platform, I think it's going to be a new world as we're moving forward. So we would, when we were particularly focused on events, we would kind of hone in on a cutoff, and if we were worried that we weren't going to fill the event, we might liberalize it, but we'd kind of try to play that, and we would fill our events. I do see some questions coming in, and I am going to touch upon these. Particularly, I see about return of results, and that's one that we could talk quite a bit about. And the registry we're using is PEER. Is the current the Genetic Alliance's registry, PEER. So, just quickly on how we process this file that is exported from PEER. We have to define the attributes we're interested in. Sometimes a multiple choice question might encompass several attributes, or it might be one attribute. It depends on our design and what we're trying to do. Single choice questions are usually one attribute. So then we'd create this table, which is basically a table that has attributes as columns and people as rows. And these attributes might be the answer to a question, or they might be something we calculate from several answers to questions, which I call derived. And then we can filter these columns and use this to filter and rank people and cut off, make our analysis that way. And that's been very effective. We've been able to use the registry data very nicely, and indeed, we've found that we've managed to hit 90% plus pass rate at these events. So then they'd come to our event. We'd have them do a consent at the event. We would require that they allow us to have proxy, which means that we can enter information in the registry on their behalf, because we chose to use a survey in the registry as our way to enter the screening data we collect when we see them at one of these events in person. And so we kind of forced the registry into this use. It's not exactly what it was designed for, but it's worked out okay. Then it keeps the data all in one place and one export. And so then we would do this clinical screening. And I'll talk in a moment about the two ways that we've done that. One we call fast track, but if we saw them in person, we would just examine them and go through a checklist and confirm that they meet the criteria. Then we would obtain the blood sample at the event, assign a coded ID to them, and record that screening data, as I mentioned. And we have a paper backup. We had events in Baltimore, New York, London, Edinburgh, Scotland, Madrid, Brussels, Rome, Nashville, Indianapolis, Chicago, all over the place. And we had more planned, and then COVID came along. And so that really threw a bowling ball into our whole method. Now, what we've been doing, we had a couple of ways to do this. We have IRB approval for method B, but we haven't used it really. So the method A is that we'd have the people. Let me back up. This is about fast track. So let me provide the correct context. What I just described was when we have somebody come to an event and we examine them. Now, the other way we could enroll someone is if we have adequate medical records to be satisfied that they meet the criteria. We might not have to go through the physical exam and clinical visit with them. So that's what now that's when I'm talking about having two methods that you might do that. One is that they can upload their records to the registry, and then we review them and complete a checklist from the records. And if we're comfortable that the clinician is well versed in this condition and has provided strong supportive clinical record, then we can do it that way. That's what we call that fast tracking them. Now, we haven't done the second method yet, which would be for a doctor in their office to do it and to complete the checklist. It's really been investigators looking at uploaded medical records to date. Now, part of our project is also going to be to integrate some existing samples or data that are out there. So I just think I should mention that, but probably no need to dwell on that too much. Well, so we were rocking along pretty well with this approach, and then COVID-19 happened. So now we had to regroup and figure out how we're going to do this. Our events stopped. We don't have our conferences anymore. We are not, and those were convenient because we were really leveraging the fact that we had clinicians coming to those conferences who would then dedicate part of their time to doing this project and screening people. And we had people coming to those conferences who were interested in participating. So now that's all sort of thrown off. And so we can't really do the screening events like we were. Should we turn to having this done within clinics? Well, that's the whole problem of the time frame of getting IRBs up and running and enrolling sites. And so we are going that direction, but it's a little bit of a slow go. So then that leaves us with medical records review, what we've been calling the fast track. And we're really thinking that that may be very productive for us. And I'll spend a little time on that here in a moment. The other side of this was that we've been sticking with blood until now, but so we thought about that we might go ahead and consider doing saliva. Saliva is really probably adequate. It's probably fine. You don't get as much DNA. It's a little bit more challenged with some of the contaminant, but it's possible to filter that out. So that is definitely an option as well. There you have the need to have the logistics of shipping saliva kits to people and having them returned. But for us particularly, it still requires that we confirm they meet the criteria for the 2017 criteria for HEDS. So we had to solve that. It doesn't solve that by itself. So now our new plan right now that we're really kind of excited about, and we're just trying to get going now, is that we're going to try to really leverage the fast track process. We have some clinicians who are in the process of obtaining IRB approval to just simply look at their medical records, which is often just a computer search, for patients who meet the 2017 criteria and have records that support it. And in particularly, one of our principal investigators, Dr. Francomano, has been fortunately has very busy practice and has been diligent about this process of recording all these criteria in a very organized form in her records, and has given it to her patients, so they have it in their possession. And so what we just need to do is notify them that they meet the criteria, that they should already have the documents that they need to upload, and that if they don't, the clinician will provide them with it. They upload the document, we do the fast track review and do a virtual consent, And now we have the issue of collecting the sample. So we've located a company called Exam One. It's owned by Quest now. And there's another sister company, Exam One Canada. In the US, they have 6,000 phlebotomists, really comprehensive geographic coverage, and they do mobile blood draws. They handle the logistics and shipping. And we found a possible solution in Europe. So we may be going to blood in Europe too, but we just found this the last day, so I don't know yet how that's going to work out. Now, once we have this data in hand and we've done the sequencing, it has to sit in one environment and reach and the software has to sit on the phenotype and genotype data to support the analysis. So that is its own next step that we have before us to settle on. So now there are a number of issues that we could talk about genetic studies, and since this question came up, I'll go ahead and hit one right now that we had to kind of wrestle with, which is the return of results. And our decision, and we spent a lot of time on this with the IRB and among ourselves, going back and forth and talking to different parties in the medical community, different geneticists. This is a very controversial area, very difficult. Where we ended was that we are not going to deliberately search for incidental findings such as unrelated sort of cancer risk, things like that. We're not going to make any deliberate process of searching for those things because that's not this research. That's not what this is about. And that's the provision of medical care that has all the incumbent issues surrounding medical care. If you do that as a planned, deliberate process, you're really embarking on a planned program of medical care. This is research. So we're not doing that, but we do know that we might stumble upon something. We might encounter something just in the course of our work. And we've made the decision that if we do, we're not keeping quiet about it. We don't feel we can just hold that to ourselves. So in our consent, we asked them whether they would like to know about these categories of items. And so we defined three categories. One is the research results, looking for some underlying cause of hypermobile EDS, new information. Of course, they all want to know about that. Second is that we come upon the fact that they have one of the known causes of EDS. Well, they typically all want to know that too, even though one of them, vascular EDS, is fairly alarming. And then thirdly was if we stumble upon something like a cancer risk. And so we asked each of these, and if they say yes, then we're going to tell them. There's some legal question about whether, if you're doing this in a non-CLIA lab, are you really supposed to release that information? But we think there's a sort of a moral obligation if you know something that you should tell them so they can go get it confirmed. So that's how we're handling it. And we are going to share information about the research results related to finding the HEDS gene. But what we share and how we phrase it is going to depend on what we find, if we find anything. So so I'm looking at some of the questions here. We do not currently have provisions for an electronic signature in our IRB approval. We are still trying to kind of determine how we're going to proceed with that. So we're going to be moving toward, as I said, medical records review and gaining consent by phone or teleconference. And whether we do electronic consent, we really have not settled yet. Electronic signature, we have not settled that yet. Right now it's no, we do it, it's always an ink signature. So yeah, so again, if we find results that are available in a lab, we're going to just tell them they need to go and have it confirmed, confirmatory test. On the other hand, you know, the new results we might find related to HEDS, if we're lucky, won't be available in a confirmatory test. So we'll just have to be careful that we provide adequate context about that. Yeah, so I think the issue of changing our mode of consent is something that we will be considering and looking at. Until this time, it hasn't been a big issue because we needed to have them come. Mostly they came and were evaluated at our events, and so we just had them sign with ink while they were there. But I think now that may work out more efficiently for us to develop the electronic approach. Other issues that you know, we had to deal with, you know, withdrawal after the whole genome sequence was performed, the European requirements about that, and who owns the data, whether we should return just the VCF files to them or not. A lot of little tricky issues to deal with. I'm going to pause and be sure we have time for other questions, because we're 42 minutes into it here, and I'm happy to spend more time on any aspect of this. So I see, can participants request or access their raw data? No, we're not doing that at this time.
Woody, I can't tell if you disappeared or I disappeared.
I'm here. Do you hear me? Let me see if my...
I can hear you.
Yes, good. Okay. Well then, I guess I'm here. So if I'm not, I guess I'll get a chat message if nobody's hearing me.
Right. So a question, so there's probably two levels of answers here for some of these questions. There's what the platform's capable of, and then what the disease advocacy group decides. So a question here, Woody, is, can participants request or access their raw data? And you can give the EDS answer and I'll give the peer answer.
Yes, so at this point, we have not, first of all, our consent does not contemplate it. We've not told anyone they would. We've not offered it. We've not suggested that we would provide the VCF files. We understand that it should be possible to do it. Right now, we're not planning on doing that. I'll just leave it there. We're not planning on doing that at this point. And we're a little bit anxious about the fact that we're not using a CLIA lab, that we've drawn these samples at diverse locations around the world, shipped them to a biobank, that we've not necessarily followed the chain of custody that you might follow in a CLIA setting. So we're just, you know, and we'd have to be sure there's malpractice coverage if there's a malpractice issue. Right now, we don't have it in the plan to be providing these VCF files to people.
Right, so now I'll give the peer answer. So peer is a research platform, and in fact, we are not a covered entity, nor are we subject to many of the usual oversight. However, we have put ourselves under all of the oversight and more. Genetic Alliance has an IRB and an ethics board, both comprised of, you can look on our website, people whose names you'll recognize as leaders in return of research results, return of results overall, use of the EHR, observational studies, et cetera. And in fact, one of the things that we're encouraging in the platform, while this is definitely a sticky issue and one that's evolving quite a lot, is that we be as transparent as possible with our participants, and that individuals do have a right to the data that is theirs. And so the platform itself does allow, for example, the upload of any sorts of genomic files from some of the common direct-to-consumer research file networks like 23andMe and Ancestry.com. Some of us, for example, my organization is uploading the research results from a 29 gene panel, and again, putting these under the control of the individuals to be accessible for research in a de-identified manner. This is definitely an interesting place, and we have had lots of discussion about it. And basically the caveat to our members is this is not a clinical exam. This is not a clinical platform. This is not a clinical result. And that if you want to take action on something you see here or your clinician does, then it needs to be verified in a CLIA approved lab, which we of course believe is very important in the clinical setting. But again, this is a research endeavor. And basically what our lawyers have told us, and again, very prominent lawyers who names you would recognize, is that in fact, there's not a legal issue here. It's really more a moral and ethical issue, and that's how we're handling it.
So, it's still kind of an open issue for us. We always could decide to make those VCF files available. But what I will say is that we did not make any promises about that or suggest that we would be doing it. So we kind of have our options open from that standpoint at this point. I think there's a little bit of nervousness among some parties on our end about it, and we probably need some further discussion.
Right. Okay. And then let's see, I'm going back through the questions, Woody, and people feel free to put more questions in the chat. If you have any, there's too many of us for me to unmute folks. Let's see, the raw data question, e-consent, we talked about that. It is, Woody has been talking a lot about our former platform. In fact, the first group moved over to our new platform that we've put together in collaboration with Luna DNA this past week. So we're pretty excited about that opening. And so Woody's been sort of straddling, as has lots of our organizations, this older portal. The newer portal has e-consent, it has messaging, a lot of the sign up. In fact, PXE International is doing sign up for clinical trial in the platform. So all of that is now possible. We're co-op run, so anything the advocacy groups need or want, we try to build. I think so far that you got all the questions, Woody, because you took a couple while we were going. One new message here. Will you return any results that may be associated with a participant's clinical features, but maybe for a different condition or diagnosis?
Yes, right. So that has these two levels. At one point, we had a plan to make a deliberate search for the ACMG59 genes. And then we started getting a little cold feet about that because we couldn't quite identify who within our organization was going to take responsibility for those results, who would be willing to be on the line for delivering those results. We became worried that these were non-CLIA results being shared, analysis. And so we ended up backing off from doing that. But then we have the issue of we still might encounter something. And so we need to contemplate that. And we felt, well, what are we going to do? We can't just sit on it if they had said they want to know. And so, yes, so when there's a question here about, other conditions like neuromuscular disorders. So I think what we would do in that regard, and what we asked in our consent, is whether they would want to know about a condition that was actionable. That's the 59, the ACMG 59. And so if we find something that's not actionable, no, we're not going to tell them about it because we didn't ask them. So no, we're not going to bring that out.
Okay, any other questions? Okay, looks like you did a good job, Woody. So I will just add that we asked Woody to present today because he's kind of going beyond just a simple registry into a study. You probably heard that he's also in the middle of what a lot of us are with regard to, so COVID came along and now how do we continue? Our webinar last week, which was on doing clinical trials differently, detailed several ways to do clinical trials at home. And so I recommend that you look at our website for that webinar as well. It gave a lot of different examples of how these things actually can be done, probably where they should have been done to start with. We're really looking a lot into how much it really affects people to have to travel to clinics, and in the very rare diseases, there aren't enough clinics or even close enough clinics to do the kind of work that we're all looking to do. So without
if I could,
Sure.
I have one quick last thought I just want to be sure I share, because I had meant to mention this, just a little small pearl, a small practical issue. If you are doing a project like this with a registry, I mentioned before to be careful about how your data comes in and that you don't have to reconcile alien data in a less than optimal fashion with your registry data. I also would say that if you're having people upload information to the registry, provide a way to categorize it. That's another thing. I meant to mention this, and I just want to be sure I share this because it's sort of been a problem for us, that people upload all sorts of things and you get overloaded with it and you can't see the forest for the trees. Or the trees for the forest, whichever. So be sure that they can categorize what they upload. So just another little practical pointer.
I think the other thing we'll mention is that in the old system, the person had to upload their own EHR and any clinical record, any gene panels, et cetera. In the new system, we have connectivity with 700,000 portals in the United States. And so the individual actually enters their portal URL, their password, and then never has to do that again unless the system, Kaiser or, whichever one is involved would change. So it's not just Epic, because Epic is only part of the portals in the United States. This is 700,000. So it's Epic, it's Cerner, it's all of the kind of homebrew ones as well. And the individual, again, enters the URL or can look up by the doctor's name. We have connectivity through the doctor or clinic's name. And the individual then can put in their password. And after that, in fact, when I wake up in the morning, I always see I have new records in my portal because I also connect my Withings scale, for example. The other interesting aspect of this system that we're now in is something that we've been dying to do since 2003 when we began our registry, and that is individuals who are part of PEER, which are now some 50,000+ individuals, receive shares in a public benefit corporation that has been set up just for this. So they're actual stakeholders, stockholders, shareholders in the company that is allowing access to their data given their permission. The data never leaves the platform. The questions come to the platform and the individuals receive shares and eventually dividends once we're profitable. They can give those shares to their organization. So if an organization like Woody's is signed up, his members could give their shares to EDS if they want to, and or to, whatever, the American Red Cross. It doesn't matter which nonprofit, and or they can keep them themselves. So what we're trying to do is really disrupt the American and international research systems from the ground up, in the sense of giving more power to advocacy communities like Woody's and like others, so that in fact those communities are in the driver's seat, the individuals are in the driver's seat, and we eventually become the hub. In my TED talk, I said we want to move the food so that cats will come to us, and we can call the shots about what are the terms that we can require. For example, data sharing, return of the results back to the platform, information found about an individual during a clinical study that are simply measures about that individual can be put into the platform. And so that essentially allows a more robust system with more complete data with us in control, and hopefully reducing the cost for everybody, for CROs, for companies, so that more research can be done on more diseases, and maybe the end result will not be astronomical prices on these drugs. So we're really trying an environmental play here in our co-op, cartel, coalition, consortium of peer members. EDS is a governor level member, so Woody and the other governors get to make decisions about where the platform will go. And we're really delighted to have such a strong and interested partner as someone like Woody, who gets it again from both ends, the researcher end and the parent end as well. Anything else? All right, well, thank you all. This presentation will be available on Genetic Alliance's website as part of our bootcamp for registry development. And that should go up in a day or two, and you'll receive an email telling you that it in fact is ready. Thanks, and tune in again for more parts of this series. Thanks very much. Thanks, Woody.
You're welcome. Bye.
Bye.